Hello viewers, today we are going to see what is nested PCR and multiplex PCR. So before going to that, we will see what is actually how a PCR is done. So in this, you will see a single stranded template and a primer is bound, and here is the second strand and another primer is bound. So here there is a reverse primer and there is a forward primer. This is how you in a normal PCR how the template and primer is binding. So in the PCR machine, you are keeping a PCR reaction conditions like initial denaturation and a cycling and final extension and holding temperature. So initial denaturation 95 degrees centigrade is for 1 minute. Then in the cycle, there is also there is a denaturation temperature that is kept from 30 seconds to 1 minute. Then annealing temperature, it is kept always 5 degree below the TM of the primer. Then again the extension temperature, and this annealing time also you can keep from 30 seconds to 1 minute. So in the extension temperature will be around 72 degrees centigrade for Taq polymerase, and for proofreading and thermostable enzymes it will be around 68 degrees centigrade. And here the extension time generally keep for 1000 base pair 1 minute, like that. And after the cycling is completed, the PCR reaction tube will enter into the final extension temperature 72 degrees centigrade. So here all the PCR products will be made up to the last base pair, and the final extension time is actually 5 times of the time what you are setting in the extension step. So suppose if it is 1 minute, you will keep 5 minutes, like that. And in the holding after cooling down to 4 degrees centigrade, you can take out the samples. So in a PCR, generally template will be used from 0.1 nanogram to 1 microgram. So depending on the size of the template what you are using. Then primer 0.1 to 0.5 micromolar, DNDP 20 to 200 micromolar, Taq polymerase 1 to 2.5 units, MgCl 2.5 to 2.5 millimolar. So this is how a PCR reaction look like. So initially you are using one copy of template, denatured, then primer annealing, then after extension you will get a products like this. So this will be entering into cycle 2. In the cycle 2 again denaturation and annealing. So the primer will bind to all 4 strands, and you will see finally 4 copies will be made. So if you see in the cycle 2, these are the suppose these are the PCR product like that. 2 and 3 are the specific products. So after cycle 2, the specific products will be increasing exponentially. So generally what you will say, the number of copies of PCR product formed is 2 raised to n copy, but actually the specific product will be 2 raised to n minus 1 copy. N is the number of cycles what you are setting up in the PCR machine. So this is a diagrammatic representation of nested PCR. So you have a double-stranded DNA and a set of primer is there, and this primer will be binding to this template. So if you see, this is one condition, this is another condition, this is a third condition, so this is fourth condition. So like that, different types of conditions you will see within the PCR tube for how the template is binding with the primer. So in some situations, what will happen? The primer and template will be a perfect base complementarity will be there, and it will bind to the exact place. And sometimes what will happen? The primer will be having slight base pairing with some other region of the template. So it will be binding with some other region, like this. Sometimes what will happen? To one template only one forward primer will bind, to one only a reverse primer will bind, or so like that there will be the primer and template will be binding at different locations, like that. So because of that, what will happen? The PCR sensitivity will be low, PCR yield will be low. So in such situations, to increase the sensitivity of the PCR reaction or to increase the specific product formation, we will use another set of primer which is internal to the primer what you have used in this step. So in this step you have used a primer region here in between this region. So you will design a primer within this like this. So first you will do a reaction using the first set of primer, then after a few number of cycles you will add the second set of primer, and the PCR will be continued. So in such a situation what will happen, even though your template is slightly degraded, or even if their primer is having binding with some other regions, your specific products will be formed from the second set of primers what you have used. So you will get a very high specific product. So this is generally used when the template is mixed with so many other template DNA, or when it is highly degraded situation, or when you expect that the template is present only in a very few copies like that. So this is the condition what we set for a normal PCR. So in the nested PCR, you can say this is one full step of a reaction. Okay. So this one what you will do is, in the nested PCR, in the initial PCR cycle, here you are using 30 to 40 cycles. So instead of that you can limit to 10 cycles. Then after that, the same program you will set it next to after completion of this. So there in the second set of reaction you will use these 30 to 40 cycles, so that the specific products will be formed in high amount. So this is how you will set up a nested PCR reaction in the PCR machine. So this is the first first reaction what you are setting up. Okay. So this is the first set of reaction. This will be completing for 10 cycles using the first pair of primers. Then after that, this will be a small volume of reaction of around 5 microliter or something like that. So into this 5 microliter, you can add another 15 microliter reaction without template. Okay, without template, only primer and the DNDP Taq polymerase MgCl2 mix or master mix. You can 15 microliter. You can add to this 15 microliter. So the next reaction in this step, this will be around 30 to 40 cycles. So here it will be using the template formed from the first 10 cycles of reaction. So because of that you will get only a specific product formed from the second pair of primers. So in this step you are using first pair of primers, and here you are using second pair of primers. So here you will not add the first pair of primers. So in this 5 microliter 10 cycle itself, the first pair of primers will be completely utilized. So this is how a nested PCR products look like in the agarose gel. So suppose if you see, are you seeing this? So here you will see multiple bands form. So the lowest band is actually the specific products, and the upper bands what you are seeing are all non-specific product accumulated in the first reaction using first pair of primers. So if your template is more in quantity, you will see products formed from the first non-specific pair of primers. And if your template is very low in quantity, you will get only a specific product formed. That is what you are seeing here. So this nested PCR actually you can use for diagnosis of samples to see whether any pathogen is present in the environmental DNA, or any pathogen is present in the sputum or blood or something like that. So you are actually using a template which is very complex, which is having mixture of different types of DNA. So among that you are going to look for one specific microorganism. So in that such situations, you can do this nested PCR. So how the nested PCR is different from multiplex PCR? So in the nested PCR, you are actually for the first PCR cycling, you are using one pair of primers. Then after that, for the second cycle of PCR, you are using second pair of primers. So these two primers are totally different, and the reactions are also done at different time points. But in the multiplex PCR, what you are seeing is you have a template. So you have different types of template in this. One template, two template, three, four, like that. So you have a different types of template. Okay, you have different types of template, or for a single template you will have primers for different regions. Okay, so this is what you are doing in the multiplex PCR. I will tell you once again. So in the multiplex PCR, either you can use one set of template, okay, one set of template, and in this one set of template itself you can design primers for different regions. Okay, forward primer, reverse primer, like that you can keep it for different regions. So this will be the reaction will be going on in a single tube, or you can have different types of template from fish, template from human, template from bacteria, template from fungus. So you can have different types of templates, and for each template you can have a specific primers. Okay, specific primers. So these are the two ways multiplex PCR is done. Either you are using a single template and different pair of primers, or multi-template and a different pair of primers. So this will be undergoing PCR reaction like we have discussed in the first instance. So here there is only one set of PCR cycling, like you have seen in the normal PCR. So and you will get a different products. So only thing what you have to take care in this multiplex PCR is the product sizes, or when you are deciding for the primer pairs, the product sizes formed should not overlap with one another. Suppose if all products have 250 base pair size, so in the gel you will see only as a one band. Even if you have four different types of DNA, or even if you are amplifying from four different locations of a single template, you will see only a single band. So you cannot judge whether all four has amplified or not. So because of that, in the multiplex PCR, the different primer pairs, when you select it, the products formed should be of different sizes. Then only you can analyze it in a agarose gel, like you have seen. So this will be from first pair of primers, this will be from second pair of primers, third and fourth, like that. So this is how you have done. So this multiplex PCR is done for pathogen identification. Suppose in a single sample itself, you can look for influenza virus, SARS virus, or you can look for adenovirus. In a single sample itself, you can look for it. Then high throughput SNB genotyping. You can use a beacon primer. So beacon primer, you can put it for different locations of a single template. So you can use for high throughput SNB genotype. Mutation analysis you can use. Gene deletion analysis you can use. Means within a single template for different region you plan primers. So wherever the gene is deleted or the DNA deletion is there, the expected size you will not see. So like that you can use. Or template quantification, linkage analysis, RNA detection, and forensic studies, you can use this multiplex PCR. So only thing what you have to take care in this multiplex PCR is you are using several pairs of primers. So you are making different product sizes of different numbers. You are making more number of PCR products of different sizes. So because of that, you have to add more DNDPs, and when you add more DNDPs also, you should not inhibit the reaction. So there is a lot of standardization is required for this multiplex PCR when you are performing in your lab. So you have to first use all the four different templates, and you have to see whether all the PCR buffer, DNDPs, tag polymerase concentration, etc. what you are using is amplifying all the four or all the different pairs of primers, then only you should go for testing it in the lab. Otherwise, it will it will give wrong errors. Means your sample will be present in the template what you have used. So because DNDP is limiting or tag polymerase is limiting, uh, the the products will not be forming. And because you are using lot of primer pairs in this reaction, so always the PCR reaction is depending on the DNDP primer template tag polymerase concentration. So you have to adjust the primer concentration also for all the primers to get a successive band in the agarose gel. So that is why multiplex PCR is you have to do a lot of standardization. Generally you can 12 to 15 PCR products you can try it, and using fluorescent labels also you can make the number more by conjugating it with different types of dyes. So this is how a multiplex PCR is working. So this is how a normal PCR, nested PCR and multiplex PCR will be differing. I will tell you once again. So in the normal PCR, you will be having only one pair of primers, and there will be only one PCR cycle. Okay, one PCR program. What you are setting up in the PCR machine. But in the case of nested PCR, you will be having one PCR cycling reaction. So in that, you will be using one pair of primers for 10 cycles. Then after that, that product will be used for another 30 cycles of reaction using a second pair of primers. And in the multiplex PCR, you have only one set of PCR cycling step or reaction, 30 cycles. So in that, either you are using a single template and multiple pair of primers, or multiple templates and multiple pair of primers. So that is how the multiplex PCR is used. So in the next sessions, I will be posting the another types of PCRs what we are commonly used in the lab. So thank you for listening. So please subscribe my channel, and please share the channel to your friends and colleagues, and also press the bell icon for the channel. So thank you very much. Thank you.